So Ros, I wonder whether you could explain what polygenic risk scores and SNP profiles are, let's say in the context of prostate cancer, your area of massive expertise.
So SNP profiles are gene variants across the whole of the genome. They're numerous, and you can type somebody for them quite easily, and then you can actually calculate a score because they're each one is associated with a small risk of a trait. And here it's prostate cancer. And then you can divide a population, say 100 men, into what we call polygenic risk score divisions, or centiles if you want to divide it into 10% centiles across the 100 men. And then you can find, say, the man at the top 98th percentile, the top 100th percentile of those 100 men, the men at the highest risk, and offer them potentially targeted screening, because if they're at higher risk of disease, maybe the screening would find more cases of disease, more likely to find aggressive disease. At the moment, this is an area of research, and at the Institute of Cancer Research in the Royal Marsden, that's the research that we're doing. But early data suggests this is a really promising approach for targeting screening in populations.
Yeah. So as I've understood it, Ros, basically it's a change in a single letter or a pair of letters that forms a SNP in a particular place, and that you find through these large studies that position increases that risk of, say, prostate cancer in that position, and you add all those different individual positions up to form that score. And through that, we found that in particular cancers, polygenic risk scores and SNPs are more important than others, haven't we? So could you share a little bit about that?
Yeah, so for example, the proportion of familial risk that's due to genetic factors is much higher in prostate cancer than, for example, in ovarian cancer, where alterations in single genes,
such as
BRCA, is much more important.
Yeah. So that in terms of your prostate, I believe, what type of level of risk would somebody on the 99th centile, when it's all added up and they're the very top highest end, what type of level of increased risk would they have of prostate cancer?
Depends on the population, but it's at least 5-fold risk, and in many cases it's nearer 10-fold.
Yeah. And I think in our study we had one patient who, to our surprise, turned out to be a 6-fold increased risk. And I think you're doing some very interesting work, aren't you? A barcode 1 study, isn't it? Can you share about that?
So that's profiling in the general population to look at, if you did targeted screening, do you find more cases, and do you find more aggressive cases within that population? And the early data suggests that it's an acceptable technique and that we may find more cases.
Yeah. I mean, as I see it, you've effectively hit in the key areas, which is trying to find those with the nastiest, most aggressive form of prostate cancer who can get the largest benefit from treatment, as compared to people who have less aggressive cancers who suffer more from the side effects compared to the gains they get, isn't it?
That's certainly the aim of the targeted screening, based on levels of risk, so that we can actually enrich the population that we find for disease that really would matter, and it really does need treatment.
So I find it very exciting that this is probably the keyest area in prostate cancer, isn't it? Finding those people who are going to potentially die of it, and then putting them through an operation which may make quite a considerable number of people impotent or give them side effects, but that you need to know that people you put through it are going to really gain, don't you?
We hope that with more modern techniques, those side effect risks will be reduced, and indeed they're already being reduced with more modern types of surgery, but there is still some risk, so we really want to concentrate on treating people that really need the treatment.
Can I ask you about SNPs in ovarian cancer?
So in ovarian cancer, the SNPs make a much smaller component of the overall risk. There is the majority of the risk, genetically anyways, due to single gene alterations. There are some SNPs now that are known to predispose to cancer of the ovary, but most of the genetic alterations that we want to find are in single genes.
So this is a fascinating area of polygenic risk scores, that in certain cancers such as ovary they form a very small contribution, prostate an enormous contribution, much bigger than the single genes such as BRCA genes, and then in breast it's about 50-50 the contribution, isn't it, of the genetic contribution to breast cancer. And so we're thrilled with the study that we've just seen the results of. Can you talk a little bit about the results that we've had of our SNP profile results?
So in some individuals we didn't have an explanation for why they'd had a cancer at young age, And interestingly, in the SNP profile results, they were in the higher in fact, they were over the 98th percentile for the polygenic risk score for their particular cancer. So we can't at the moment prove it, but it is tantalising that that might actually have caused their early onset of disease. We do need more research to prove this in larger cohorts, but it is looking like when you're in the higher decile of the polygenic risk score, that you really are at higher risk, and that we are enriching for more aggressive disease, sometimes at younger age of onset as well.
Yeah, I think you pretty well lost your breath when you saw some of the results, wasn't it? They were so extraordinarily poignant and hitting exactly people who we knew had some reason for what they'd had. And so essentially when we do genetic screening, the key is, isn't it, to have doing the single high-risk genes, but to do the SNPs as well, isn't it?
That's the nice thing about the whole genome programme, is that you get everything all in one test.
Yeah, and the pharmacogenes and the recessive genes, yeah.
Yeah.
Thank you, Ros, very much.
Thank you.